We will present this work with Isabelle Verdiez-Metz, and it's a work that has been achieved in the frame of the PhD of Marie Fretin. We have some interesting results in this experiment concerning the low-input mountain farming systems. We designed the low-input farming systems, and we studied the impact of these systems on cheese sensory properties. I will present the result, and also on teat, milk, and cheese microbiota, and Isabelle will present these results. So, some words about the context on milk production in mountain areas. Well, due to high cost and low productivity of the cows in these systems, mountains cannot compete. And in particular, in the context where there is an increasing price of inputs, mainly of cereals, because cropping is not possible in these areas. So the farmers, one strategy is to try to maximize the use of local resources, namely forages, and also to reinforce the link to terroir. So mountain areas have also to capitalize on their strengths, and one of their strengths is their natural image and the very high quality of local products and PDO cheese in particular. So in this context, we designed two innovative low-input dairy systems. You will see that they are very low. They were, in fact, two small farms within the same experimental farms. They were completely separated. So we had 12 Holstein and 12 Montbéliard cows and heifers that were reared in each of the low-input systems. And in order to maximize the production on pasture, the calving season was in early spring. And this calving season was very short, only 70 days. 70 days for the reproduction. We also complied with some specification of the PDO cheese that are in this area, Saint-Nectaire and Cantal in particular, so no use of silage. And this experiment was a long-term experiment. It lasted for 5 years. So we had two farming systems. Some characteristics of the farming system. The first one was called extensive. You can see that is very extensive because it was a zero-input farming system. Zero mineral fertilizers, zero concentrate, low stocking rate. So 24 cows reared on 60 hectares of permanent grassland with a very high biodiversity, late mowing, long rotational grazing. And this system was compared to another one that we called semi-intensive. Semi-intensive for this area, we could call semi-extensive or extensive also. 30 hectares of old temporary grassland, minimal amount of mineral fertilizers, 4 kilograms per day of concentrates during 150 days, and a lower biodiversity, but they were all temporary grasslands. So we tried to make functioning these two farming systems. So some results that we had in this long-term experiment. One of the aim was to maximize the production on pasture. We could do this because we produced between 75 and 80% of the milk on pasture. But as you can see there, for semi and ext, the milk production was very low. The peak was at 22 kilograms per day and per cow. So in average, in the extensive system, we produce 4,100 kilograms milk only. And in the semi, it was a bit better, but not really higher. And the efficiency of the concentrate was not very good in these semi systems. In the semi system, we have a better persistency, but so this average production is very low, but it's the the average of the total milk that was produced by the two groups of cows, divided by the number of cows. And we had some problems with the reproduction, so some cows were dried and so on. So this average also takes into account these problems that we had. So as we make cheese in this area, one concern was the quality of the milk of these seasonal farming systems. And you can see here that there is a period in summer where the protein content is very low and is below the threshold of 30 grams per kilograms. So one of the questions was, are we able to make good cheese with these milks? So the objective of the result that we present was first to check if it was possible to make cheese with these milks, the first one, and we wanted to also to compare the sensory properties of the cheeses resulting from the two farming systems according to the milk treatment. So I will present the result. And the second objective was also to study the bacterial community assembly from teat skin to ripened cheese. And it was very interesting in the frame of this experiment because it's a long-term experiment and because the microbiota at the farm level had time to stabilize, and we wanted to take the opportunity to work on this question in the frame of this long-term experiment. So the experimental design for the cheese production, we chose two grasslands, quite extreme, a very biodiverse grassland that you can see here in Ext, 86 different species, and we compared this to a more conventional grassland in this area from SEMI. It was, we had anyway 36 different species. We made cheese at two periods, in July and September. We had raw milk and we produced raw milk and pasteurized milk, and we had 3 replicates. So in total, we had 24 Cantal cheeses made with unskimmed milk in our experimental dairy plan, and they were aged for 5 months. So we made sampling and analyses on the grass, on the milk, and of course on the cheese, regarding fatty acid, milk composition, grass composition, sensory analysis, and so on. So the results concerning the milk first. In this experiment, the milk that we use for the cheese making, you have the gross composition that is reported here. You can see that in the SEMI, we had lower fat content. This is due to the low fiber and the concentrates that were given to the cow. There was some problem with the subacidosis in this farming system, and we had very low protein content on Ext farming system, which is linked to the low digestibility of the grass and the absence of concentrates. We had also a bit more somatic cell counts in the SEMI. It was observed during all the duration of the experiment. And some results concerning the fatty acid. We had quite important differences in the composition of the fatty acids that were linked, in fact, to the high biodiversity and the fact that we had zero concentrate in the Ext farming system. So we had higher oleic acid in the Ext farming system and also higher omega-3 fatty acids. And we also observed some differences for the classical bacterial analyses that we made. We had a bit more lactic acid bacteria in Ext and also heterofermentative facultative lactobacillus. Concerning the cheese now, we had a very important effect of the farming system on the cheese texture. This effect was mainly linked to the fat in dry of the cheese. The cheeses in the Ext system had a higher fat content. They were more creamy, and more adhesive. on the sensory analysis. So they are linked. In fact, the difference are linked to the milk fat to protein ratio and also to the fatty acid profile of the milk. They are quite classical effects. We also observed some differences in the yellow coloration of the cheese that were due to the content in carotenoids, and in the semi-farm we had a higher amount of carotenoids due to the grass vegetative stage. It is quite classical also, and we were quite surprised because concerning the cheese taste and flavor, we had some differences, but they were quite limited. For instance, we had the cheese from the extensive grasslands were a bit more sour. We had also differences in proteolysis, and also the volatile fatty acids were higher in this system, but the consequences really on the cheese sensory properties were lower than expected. Some words about the influence of the pasteurization. Conversely, the influence of the pasteurization was very high, and heat treatment had a high impact on cheese texture. There are very classical results. Pasteurized cheese were firmer and more elastic due to a really slower proteolysis. One thing that maybe is a bit more surprising is that pasteurized cheese has had really a thicker rind, a more developed rind. We think that it's due to the better development of ripening starter due to lower competition with the native flora, microflora of the milk. And concerning flavor, we had also an important influence. We had less intense and diversified flavors, and due to a slower proteolysis and a slower production of volatile fatty acids. One key question that we wanted to address in this experiment were the interactions between farming system and heat treatment. In fact, our question, scientific question, was mainly, are the difference between the farming system, according to the farmers' practices, in fact, observed only in raw milk? And we could find some of this interaction. You can see here, for instance, that with the raw milk cheeses, we had a higher proteolysis on the extensive system, more intense taste, more intense fermented cream area, and as we were expecting, with the pasteurized milk cheese, this difference completely disappeared. So it means that we confirmed that there are interaction, but the previous results that we had with Isabelle were only partly supported by our results, and in fact the reason is mainly that the two grazing systems, okay, they are different, but they are not extreme, and maybe they were too close to exert really a significant effect. So just to summarize these results, we can say that the heat treatment has really a major influence on the sensory properties of the cheese, very classical results. The grazing system has an important influence on milk composition and cheese texture, but limited influence on cheese flavor. This influence was higher in previous results, and the interaction between heat treatment and grazing system was confirmed. So we can say that indeed heat treatment limits the difference between the grazing systems, and it raises an important question to my point of view, is that the link to terroir, we can say with it that the link to terroir is weaker when milk is pasteurized. So we have achieved maybe 3 or 4 experiments with this aim. In this case, again, we confirm this. Okay, so now Isabelle will present the results on metabarcoding.
Good morning. So The second objective of our study was to explore the bacterial community assembly from coated skin to ripened raw milk cheeses, and to test if the hypothesis that our two contrasted grazing system could serve as a lever to shift the microbial community from the raw milk and the cheese via the teat skin microbiota. The factors that contribute to the construction of cheese's microbiota have become subjects of interest for researchers studying cheese microbial ecology. The community of the teat skin depends on the dairy farm environment. The microorganisms can colonize the teat skin because they're in contact with the bedding system. Bedding system, that's a factor that influenced by feeding system and housing conditions. And the feeding system is known to affect the microbial community of the rumen and the faces are also in contact with teat skins. The teat skin is considered as a major reservoir of the microbial diversity of raw milk because many bacterial genera found in raw milk are also detected in teat skin. And the teat skin is inhabited also with many microorganisms that also grow in cheese during ripening. And the teat skin is also affected by many hygienic practices. The composition of the raw milk microbiota depends on that of several reservoirs, such as teat skin, but also milking equipment, including milking machine, pipelines, and bulk tanks. In addition to starters and the ripening cultures, the cheese-making equipment and ripening equipment and environment also influence the microbial compositions on cheese ripening. The microbial composition of raw milk play a major role in the development of the flavor in cheese. In our study, as explained Bruno, we made a lot of analyses, but I only explain this one. Of course, Susan Jordan explained very well that yesterday. And so the the bacterial population of the teat skin on the raw milk and the cheeses were investigated by using the metabarcoding. So we obtained profiles of bacterial diversity, and these populations are represented by the OTU that groups the DNA sequences according to their homology. The bacterial populations at phylum level in our 4 habitats, we studied the teat skin, the raw milk, the cheese core, and the cheese rind. And we can see that the predominant phylum in the all habitats were Firmicutes. And the second one was the actinobacteria, and the actinobacteria decreased, relative abundance decreased from teat skin to cheese rind, and there are higher abundance in cheese rind than in cheese core. The last one was the Proteobacterium phylum. It was very abundant in raw milk, probably due to the storage temperature and storage duration of the milk. In more details, we can see in total, we can find 365 OTUs in all habitats. And we present here the relative abundance of the 34 OTU present at more than 1% in at least one of the habitats. We can see that the bacterial diversity decreased from the teat skin to the rind. And we can, in small details, in the teat skin we found 300 OTUs, and the majority were assigned to the genus or to the genus of Corynebacterium. 3 other OTUs, also with higher abundance on the teat skin, were also detected in the milk. They were assigned to Diatsimaris, Turicibacteria, and Robustia. The lactic acid bacteria, such as Lactococcus and Lactobacillus, were in higher abundance in all the dairy products, raw milk, quart cheese, and rind cheese. And in addition, the other important microorganisms on cheese rind were assigned to ripening bacteria, just as Brevibacterium, Nocardiodis, and Brevibacterium linens. In this graph, we represent a network analysis, and it was to show the flow of the microorganisms from teat skin to rind. We can see, as said before, that the teat skin present the higher bacterial diversity. You will see here that the white nodes represent the OTUs, and the size of the node were proportional to the lines they reline the different nodes. We can see that 85% of the bacterial species present in the mix were also detected on the teat skin, and 27% of the bacterial species present in cheese, quart and rind, were also detected on the teat skin. In more detail, we can see with the Venn diagrams. The Venn diagrams indicate the occurrence of the 365 OTUs across the habitats from teat skin to ripened cheeses. According to the 300 OTUs detected on teat skin, 1/3 were also detected in the dairy products, especially in raw milk. And these OTU were included bacteria of technological interest, such as Brevibacterium linens, Arthrobacter, Corynebacterium, and Lactococcus lactis. This confirms that the teat skin is a major reservoir of microorganisms for raw milk. 1/4 of the OTUs detected in the cheese, quart and rind, were also detected in teat skin, and they were assigned to bacteria potentially involved in the development of the flavor, the aroma, and the color of the cheese. They were assigned to Brevibacterium lemons and Staphylococcus aureum. For the first time, we can see that the teat skin could be the secondary reservoir of microorganisms for raw milk cheeses. The majority of the OTU detected on the quart cheese were also detected in the rind. 76 of these OTU were assigned to Lactococcus lactis or Lactobacillus, and 22 OTUs were exclusively detected on the rind, and they were assigned to ripening bacteria such as Brachybacterium, Brevibacterium, and Nocardiopsis. It's probably due to the fact that the rind is an open system in contact with the environment, as explained Benjamin Wolf yesterday. Finally, only 9 OTUs were found in all habitats. They were assigned to 7 genera, and most of them are involved, important for cheese making, such as Lactococcus lactis, Lactobacillus, and Streptococcus. So, what is concerned the grazing system? We can see we are represented here the distribution of the sample according to the relative abundance of bacterial species. And we can see first that the composition of the teat microbia is very different from that of the microbiota of dairy products. And according to the grazing system, we can see that we can well separate the two teats. The teats of the crow reared in the semi-grassland where majority we found majorly Brevibacterium, Bifidobacterium, and Clostridium. Conversely, the teats of the ex-crows were dominated by Corynebacterium. And we can see that the low effect of the grazing system on the microbiota of raw milks and raw cheeses. When we see the major effect was due to the two system grassland system where affect the teat skin, and we try to explain this effect. Probably the high diversity of the grassland, of the ex-grassland, and the high fertilized grassland, and the distribution and concentrate could change the microbial abundance, diversity, and activities of the bacteria on the soil surface and in the phyllosphere, where the cows are breeding. But we cannot. The link between the phyllosphere and the soil surface and the teat microbiota is not yet well understood. Another effect was that some of cows in semi-grassland were in subacute ruminal acidosis, probably due to the daily distribution of concentrate, and this affects the microbiota of feces and so the teat skin microbiota. On the teat skin microbiota, we also observe more higher abundance of some species such as Atopobium, Brevibacterium pseudolongan, and Turicibacter, and lower dominance of Solibacillus. And so these results are in accordance with those of Mao and al. In the rumen and in the faces. In conclusions, we can see that the teat skin was the primary source of microorganisms for milk, because 3/4 of the species detected on the teat skin were also found in milk. And teat skin is also a potential secondary source for microbial diversity in cheese, because at least 1/4 of the OTUs on teat skin were also found in cheese. Concerning the grazing system could be a key driver or teat skin microbiota assembling and of milk and cheese chemical composition and texture, but it had a low impact on milk and cheese microbiota and cheese flavor. The explication of some of them that said Bruno that our grazing system were contrasted but not extreme. The animals were reared in the same farm, the same environment and same milking machine. And the cheeses were also made in the same plant with starter, and the level of the starter could be lower to see perhaps some more differences. So now it could be interesting to have further studies from the dairy environment, from the soil and the grass, the microbiota of the grasses, to cheese, and not in just one plant, but in several formats. So these results are published in 2 articles that you can find, and we want to thank all of the contributors of the study, and thank you for your attention.